My name is Sergio Lozano. My partners are Eros, Aranda, and Alondra Curiel. Today we'll be talking about immunologic tolerance, autoimmunity, and hypersensitivity. So how does the immune system maintain unresponsiveness to self-antigens? The immune system maintains unresponsiveness to self-antigens through a process known as immune tolerance, which is known to occur at both the central and peripheral sites. Central tolerance takes place during the first steps of bone T and B cell development, which happen in the thymus and bone marrow. This is where the cells that strongly bind to self-antigens are eliminated through apoptosis or corrected through receptor editing. This isn't always the case, as some self-reactive cells evade the process, which leads to peripheral tolerance as a second backup. Self-reactive cells in peripheral tissues are either rendered anergic, suppressed by regulatory T cells, or killed through apoptosis. Inhibitory signals delivered by molecules such as CTLA-4 and PD-1 also help to weaken the immune response to itself. These measures prevent autoimmunity and help maintain the body's non-self discrimination ability, keeping the immune system in balance so that it fights external pathogens without harming itself.
Now, what are the factors that may contribute to the loss of self-tolerance and the development of autoimmunity? The loss of self-tolerance and the eventual development of autoimmunity are two factors that work hand in hand with one another. A combination of genetic, environmental, and immunological factors contributes to the loss of self-tolerance. Certain genetic mutations, specifically in the HLA coding region of DNA, can significantly impair the body's ability to develop self-tolerance. Environmentally, triggers such as infections, drugs, and other toxins stimulate abnormal immune responses and even modify self-antigens. Finally, some immunological processes such as inflammation can cause the release of self-antigens and lead to the recruitment of antibodies against these antigens. All these factors lead to a loss of self-tolerance and a buildup of high-affinity self-antigen antibodies, which ultimately contribute to the development of autoimmunity.
What are the mechanisms of different types of hypersensitivity reactions? There are 4 different types of mechanisms. The first mechanism is type 1, also known as the immediate hypersensitivity mechanism. During the sensitization phase, the initial exposure to an allergen triggers Th2 cells, which will stimulate B cells to produce IgE antibodies, and then these IgE antibodies will attach to mast cells and basophils. When there is re-exposure to the allergen, it will stick to IgE on the cells and cause them to release histamine, prostaglandins, and leukotrienes. The chemicals cause blood vessels to widen, fluid to leak out, and mucus to be produced, leading to allergy symptoms like runny nose, asthma, or even anaphylaxis. Type 2, also known as antibody-mediated cytotoxicity, is when the immune system makes IgG or IgM antibodies that will attach to antigens on the cell's surface or in the extracellular matrix. The binding will activate the complement system, causing cells to break apart and die. This can also lead to opsonization or ADCC, where natural killer cells will kill cells coated with antibodies. This process will cause tissue damage and lead to diseases like hemolytic anemia and Goodpasture syndrome. Type 3, also known as immune complex mediation, will form antigen-antibody complexes in the blood. These complexes can get stuck in different tissues like blood vessels, kidneys, and joints. When the complexes build up, it will activate the complement system, attracting neutrophils to the area. The neutrophils will then release enzymes and chemicals that cause inflammation and tissue damage. This can lead to diseases like lupus, serum sickness, and certain types of arthritis. Type 4, also known as delayed type hypersensitivity or DTH, is where the reaction is caused by the T cells instead of antibodies. When the body is exposed to an antigen, Th1 or Th17 cells will be activated and chemicals are released that bring macrophages and neutrophils to the area, causing inflammation and tissue damage. CD8 lymphocytes can directly kill the body's own cells that display that antigen. It is called delayed type because it usually takes 24 to 48 hours. Some examples are contact dermatitis, which can be caused by poison ivy, TB, and type 1 diabetes.
That'll be it. Thank you and Kegum.